COVID-19, a comparison of species bonded by spike proteins, presented by Margaret Lund, Kylie McCann, Sammy Morgan, and Zachary Oshiro. The introduction column gives background information on SARS-CoV-2. The image labeled SARS-CoV-2 describes the structure of a COVID-19 molecule. Take note of the spike glycoproteins. COVID-19, also known as SARS-CoV-2, is an invasive coronavirus that has infected millions around the globe since its discovery in December of 2019. SARS-CoV-2 is a part of the beta coronavirus genus and is the most transmittable among its kind. Others of this family include SARS-CoV and MERS-CoV. The spherical cell of SARS-CoV-2 consists of spike glycoproteins, M proteins, E proteins, and within its viral envelope exists the nucleocapsid protein and RNA. Using these structures, primarily spike proteins on its surface, SARS-CoV-2 can enter the cell of the human body among other species to initiate widespread cell invasion. SARS-CoV-2 is most transmittable through the nasal cavity, eyes, and oral cavity through droplets from respiration. ACE2 receptors latch onto the host and invade them. Then the virus multiplies. By comparing various species to humans, scientists can predict how likely COVID-19 can transfer from one animal to the next. Since a bat was the primary transmitter of this virus to a human, it can be assumed that bats and humans have a very similar critical amino acid sequence in their receptor binding domains. This is because if an animal virus can invade human cells, its receptor binding sequence must be very similar to the human virus receptor binding site through the comparison of the critical amino acid sequence of humans. Pangolin, European mink, tiger, Asian palm civet, and bat. Scientists can identify if this prediction holds true.
Discovered for our materials and methods is that the National Center for Biotechnology website contains an extensive amount of information that has tracked various viruses and their elements. This website allows users to search among these viruses to track its origin and evolutionary patterns, as well as the epidemiology of said virus. Nucleotides and the coding of proteins are also included within the NCBI. When searching the SARS-CoV virus, data about the virus is listed. This website also lists a variety of strains of the SARS-CoV virus. Each strain has a protein sequence which makes it different from another. This sequence allows users to analyze amino acids along this chain of capital letters. The single letters shown along the top of the screen may align with a green-colored triangle. This triangle indicates that the amino acid capital letter shown is a part of the viral sequence of the S protein found on the surface of the SARS-CoV-2 cell. By comparing strains of the SARS-CoV virus, researchers can further understand the complexity of SARS-CoV-2 among other viral strains. Our references are listed below the box that shows the results.
So I'm going to go over the results portion of this project. If you look at table 1, you can see that the species we chose to compare to humans in regards to their amino acid sequences are pangolins, European minks, tigers, Asian palm civets, and bats. What we found when we compared their amino acid sequences was that between humans and pangolins, the percentage similarity was 70.6%. Between humans and the European mink, it was 88.2%. Between humans and the tiger, it was 94.1%. Between humans and the Asian palm civet, it was 47.1%. And between humans and the bat, it was 17.6%. And then below our percentage similarity results, you can see a legend that refers to the single letter amino acid codes. So pretty much telling what these capital letters mean, like what amino acids they correspond to. And if you go back to table 1, you can see that these numbers on the numbers on the side with the plus 11s are there to represent the location of each sequence.
The discussion section describes what a zoonotic disease is, such as COVID-19, further explains results, and discusses how COVID-19 can be further studied amongst the scientific community. To begin, when SARS-CoV-2 was first identified amongst the science community, researchers had to figure out where this destructive virus came from. It was brought to the public that this disease had originated from a bat and had spread to humans among other species. This is known as a zoonotic disease. A zoonotic disease is an infectious disease which is caused by a pathogen. This comes from the source Zoonotic Diseases from 2017. The next few paragraphs explain modes of transmission, likelihood of transmission, and why a bat may have been the primary transmitter. To further understand how SARS-CoV-2 became the viral disease it is known as today, more research must be conducted. More species must be tested to see the connection from one species to another. One must consider how a species has gotten it from another. Was it through close contact, eating the same food, saliva, urine, or excrement? Although SARS-CoV-2 now consists of more variants, researchers must first understand the source to a great length to speculate how this disease may affect life in the future, near and far. And that concludes this presentation on SARS-CoV-2, COVID-19.